And now the flash talks. Thank you, guys.
So aging affects each organ at a different pace, and understanding how these variations could redefine, they could redefine how we detect and treat age-related diseases. So we went to UK Biobank, and we used the plasma proteomics of about 50,000 participants, and we built both first generation and mortality-based second generation organ-specific aging models. And our models show that aging in organs, accelerated aging in organs like lungs or liver, strongly correlates with diseases in those specific organs or systems. And they suggest that chronic diseases are actually manifestations of accelerated organ aging. And then we went on and we showed that sex, lifestyle choices, diet, and even certain medications can affect aging across different organs in different ways. And the models are also interpretable. Okay, so poster 4, if you want candy.
Yeah, I'm Albert Ying from Harvard Medical School, and we're in the past year we're building this platform called BioLearn, which we're showcased that we can use them to benchmark and evaluate aging biomarker in various data sets. And later I will also have a talk. I will explain more. And also, if you catch me and if you want to talk more about my other project, we have a causality clock, we have a clock-based website, we have a MesoGPT, which is a foundation model for DNA methylome, and we also have AGM, which is a high-dimensional representation for aging. So if you want to discuss any of this, feel free to catch me at my poster. It's in number 6. Thank you.
Hi, my name is Alexandra Binder. I'm an associate professor at the University of Hawaii Cancer Center, and my poster is on the use of epigenetic clocks to predict chemotherapy tolerance and use as a way to appraise interventions to mitigate aging over chemotherapy. So as we know, chemotherapy is not always well tolerated, and dose modifications may contribute to worse prognosis. So we conducted a clinical trial of stage 2 and stage 3 colon cancer patients assigned to usual care or resistance training intervention. And we saw that specifically GrimAge at the beginning of chemotherapy could be used to predict dose modifications. We saw epigenetic aging over chemotherapy, as others have seen as well. And also that our intervention mitigated epigenetic aging specifically in the GrimAge clock. So if you're interested in this and hearing more about this study, please come visit me at poster 8. Thank you.
Hello, everyone. My name is Suri Zhang, a postdoc in Vadim's lab. When we discuss aging, we always talk about methylation, gene expression, protein level, but there is still a very critical process, which is kind of missed: alternative splicing. There are a lot of alternative splicing events changed in aging, which is not a surprise, but here we focus on the alternative splicing events that could cause some damaging consequence on the protein function. Using this kind of events, we quantified the splicing degeneration level, which could be served as a reliable biomarker of aging. We also did some research on the mechanism of aging-related splicing degeneration. If you are interested in it, please visit my poster number 10. Thank you so much.
Hello, everyone. I'm Francesco Morandini, and transposable elements are repetitive sequences that are able to self-replicate in the genome and cause damage. They have been shown to activate during aging and disease, and so we asked if we could make a biomarker out of them. We found that CPGs in transposable elements are particularly susceptible to losing methylation during age. And we used either the methylation of individual transposon copies or their genome-wide average methylation state to create chronological age biomarkers. Finally, we investigated associations between hypomethylation at transposons and various diseases, and we find that ancient inactive transposons lose methylation throughout your entire lifespan, but are not associated with disease, while young active transposons only de-repress when you're over 65 and associate with disease. Come to poster 14 to learn more about this.
Hi, everyone. My name is Lucas. If you ask someone who works with single-cell RNA sequencing, they would probably describe a cell as a bag of RNA. Sort of an alternative way of thinking about this is a cell as a bag of 2001 base pair DNA fragments with a cytosine in the middle. So with this approach, I developed a model called CPGPT. So it's CPG pre-trained transformer, and it's a foundation model for DNA methylation. It can do many things. So it can do genome-wide imputation, array conversion, and a lot of other things. So if you want to learn more how a large language model can understand epigenetics, come to poster 16. Thank you.
Hi, everyone. I'm Jay Chandra. I'm a medical student over at Harvard Medical School, working under Dr. Vineet Raghu and Dr. Michael Liu over at MGH. Our group previously created a CXR-based biological age, and the purpose of this work is to compare the CXR-based biological age to existing markers of biological age, specifically based off of epigenetics. And we do this using a new data set called the Project Baseline Health Study that has data from over 2,000 patients from 4 different sites in the US that has DNA methylation data, chest x-rays, transcriptomics, proteomics, and a vast variety of other data. And the basic results is that the chest x-ray based biological age better predicts or is better associated with cardiopulmonary function than these existing epigenetic ages. Thanks.
Poster 20, my voice clone will tell. We developed a clock specifically optimized for Japanese individuals. Like existing clocks, ours shows factors such as smoking, drinking, and lack of exercise accelerate aging. Interestingly, clocks like Pavlov clock and PC Fellow-Hodge incorrectly link moderate exercise with accelerated aging, highlighting the need to tailor clocks to different racial groups. By applying a new machine learning approach called Transfer Elastic Net, we integrated Japanese DNA methylation data, significantly improving age and phenotypic age prediction accuracy for Japanese individuals.
Hi, my name's Sam. I'm a PhD student in Tamir Chandra's lab. With my poster, we wanted to answer 2 main questions. So the first was, can we explain all the different types of age-related CPGs with a single model? And secondly, can we explain the scaling of methylation rates of maximum lifespan across mammals that we saw in a previous study? And so to do this, we created a model of how methylation changes occur and are propagated via stem cell division. And we fit this to data from humans and various other mammals. And as for the results, please come and see the poster. It's number 24. Cheers.
Hi, my name is Shain Rechsteiner, and I'm happy to present you the ATA clock, an aging clock based on chromatin accessibility. To develop the clock, we generated a large aging data set with data from RNA sequencing, ATAC sequencing, and flow cytometry data from healthy individuals spanning a broad range of different ages. We surprisingly also saw that chromatin accessibility relates to changes in gene expression. So our ATA clock is more interpretable than standard DNA methylation clocks. Then when we look at the changes in chromatin accessibility in more detail, we see that sites that open with age are related to inflammation, whereas sites that close with age are related to heterochromatin maintenance. And then finally, we also show that chromatin accessibility is able to predict age, and it does so better than gene expression. If you want to learn more, please visit me at poster 28.
Thank you.
Hello, everyone. My name is Xiaotao Shen. I'm from NTU Singapore. And so today, the talk is about the nonlinear dynamic of multi-omics profile during aging. So we want to know how people age, linear or nonlinear. So we just use a cohort contains 108 participants, and we collect the different OMS data. So you can see from the figure, the left figure. And we found that most of the molecules and microbeads, they are nonlinear change during human aging. And then we also use a method, we found that there are two waves of the aging. And so you can see that this is not only for all the molecules, for the protein or the metabolite, there are, the nonlinear change for all the molecules. So you can see. And so we also found some of the pathways and the modules, function modules associated with the two peaks of this aging. So if you want to learn more, come to the poster 26. Thank you.
Hello, I'm Chong Ho Lau from Imperial College London. I work in metabolomics and aging. So there are currently 3,400 metabolites, which is detected and quantified in the human metabolome database. And many of them play a very active role in regulating our body physiology. Despite that, at the moment, it's still quite hard to find a clock that will reliably predict chronological age using metabolomics across multiple cohorts. In this study, using mass spectrometry-based blood data collected from 2,000 individuals across two study populations, we're able to train a clock that will predict chronological weight relatively well. But importantly, we're also able to check and show that the change in the predicted age is strongly related to the change in chronological age using follow-up sample in the data set. And also, we're able to show that the predicted age gap is associated with mortality, physical function, as well as cognitive functions. Thank you very much.
Hi everyone, I'm Wei from the University of Washington, working with Professor Sun Li. And today I'm presenting a new AI framework called StressVI, which is designed to isolate the aging-related signals from the single-cell RNA-seq data. And as you can see from the upper right figures, so in the first set of figures, we learned the global aging patterns that are shared across all of the tissues and cell type pairs. And in the second set of figure, we learned these local aging patterns that are specific to each tissue and cell type pair. And we also apply the XAI, explainable AI methods to identify the aging-related genes from the model. And please come to my poster, number 32, if you're interested in how the model works and the new biological findings we can make. Thank you.
Hi, everyone. I'm Tsuyoshi, come from Japan. We created new epigenetic clock models by combining existing clocks. We use algorithms proposed in our previous Nature paper in 2022 on stroke genetics. And the new clock is modeled as a linear combination of existing clocks. And we applied our model to the biomarkers of aging challenge of the phase 1 and phase 2. In the phase 1, our models were ranked as 7th. And for the phase 2 mortality prediction, the ranking 6 is at the stage of this slide creation. And finally, at the submission close, we ranked for the mortality prediction based on proteomics data, number 1. Thank you.
Thank you.
Hey, good morning. I'm Max Unfried from National University of Singapore. I've been working on lipid clocks, and I want you to take 3 things with you. A, lipid clocks work. They can predict biological age. They can predict survival of cancer. What we did, we trained it actually in pathological data. So we can actually use data of deceased people and still build reliable clocks from them. That opens up like new big data sets from cancer and other diseases. And what we see is like that swing lipid metabolism is actually like the largest driver of this clock. I'm at poster 35, so come by and find me. Thank you.
Hey everyone, I'm Pete Mullen, an assistant professor at USC in Los Angeles, and my lab takes a systems bios approach to describe how metabolism changes in different organisms. I've just put a few of them there, in different organs, in male and female, and then we do untargeted and targeted mass spec, and we use that data to find metabolic changes by age, but also by sex in individual organs, and then we work with Brian Kennedy in Singapore to develop metabolic clocks, which will then tell us maybe key driver metabolites. for each organ. So if you want to find out more about that, come and chat to me at poster 36. Thank you.
Hi, everyone. My name is Sahar Daradia, and I'm an epidemiology PhD student at UC Berkeley. Epigenetic modifications are involved in the onset of cardiometabolic disease, yet existing research has focused on adults, which overlooks the early life origins of these conditions. So we analyzed data from the Chamacos cohort, which is a study of Mexican-American children from primarily low-income and agricultural backgrounds. We measured DNA methylation repeatedly in these kids from birth to age 18 and used this to estimate a novel measure of cumulative epigenetic aging. And what we found was that accelerated cumulative aging was significantly associated with several adverse cardiometabolic outcomes at age 18, including higher BMI, higher pulse, higher blood pressure, waist circumference, and body fat percentage. If you have any questions or if you would like to learn more, you can stop by our poster, which is number 38. Thank you.
Thank you.
Jolin Kuo from University of Connecticut Health. My group has used the UK Biobank plasma proteomic data to train a predictor for mortality. This poster is about the second one to predict the health span. By definition, time to the development of a major chronic disease or death. In addition to traditional the validation, we got to compare these two predictors. We look at the, if there's any benefit using both predictors, then individual predictors. Ongoing work includes GWAS on both predictors. We also have an ongoing project looking at the biological aging and major depression disorder. So I have more to chat about. You are welcome to start by number 40.
Hi, my name is Cecilia Magalhães. I'm a postdoc in Gladshap Lab, and I'm interested in investigating the microglia role in brain aging. Microglia is a long-lived cells resident of the central nervous system. It's quite important in the young brain. However, in the old brain, microglia become pro-inflammatory and also quite dysfunction. So what if we just deplete the microglia from the old brain? If you're interested in discussing this approach with me, please come find me at poster 42. Thank you.
Hello, my name is Tao Huyn. I'm a field application scientist at Biomodal, and I'm here to share with you the benefits of the 6-base genome. The Duet multi-omic solution Evoke gives you both combined epigenome, genome, and methylation information, including all 4 canonical bases, as well as 5-methylcytosine and 5-hydroxymethylcytosine. With this information, you can determine somatic versus germline mutations, methylation status of different control elements, differentially methylated regions, variant-associated methylation, as well as cfDNA fragments profiles. You can also use machine learning to predict gene expression, chromatin accessibility, and different enhancer states. All of this information is possible with as little as 5 nanograms of DNA. Conventional workflows require multiple assays and a pieced-together pipeline, but we offer a single streamlined solution. Please stop by our booth if you'd like more information. Thank you.
Hello, I'm Brianna Stubbs. I'm from the Buck Institute for Research on Aging, and I'm interested in ketone metabolism and aging. So for those of you who don't know, ketone bodies are produced naturally during fasting in a ketogenic diet, and they function as an energy, but also as a signaling metabolite. And those two roles intersect with multiple hallmarks of aging, and that's what makes ketones an interesting possible gerotherapeutic. Ketone esters are drinkable ketones that induce ketosis without any other dietary changes, so you can keep the sneaky slice of pizza on the menu, but they've yet to be tested in older adults, which was the aim of this study. The study focused on safety and tolerance. However, we did collect biospecimens both before and after our 12-week intervention, and we're in the process of analyzing the results. And if you want to see what we have so far, come to poster 46. Thank you. Thank you.
Our main target, main target of our research is longevity in the centenarian. So we have already recruited over 1,000 Japanese centenarian whose average entry age is 106. So we also have another aging cohort, healthy ages, who are 85 to 89, and they keep independence at entry. So we are analyzing longevity factor and biomarkers found in the centenarian against healthy ages to confirm significance and to assess how longevity factors works. So in this presentation, I will present the genomic component of the centenarian. Genetic component of centenarian is associated with the end of the health span, age at end of health span in healthy ages. Thank you.
Hi, my name is Owen Ham, and I'm a PhD student from Karolinska Institute. And in this study, we analyzed the IgG glycans in a Swedish longitudinal cohort, and we calculated a marker called the glycoage to integrate the age-related changes in those IgG glycans and estimate a trajectory for the glycoage. We found that the glycoage increases with chronological age towards a more pro-inflammatory state. And the glycoage predicts a higher risk of all-cause mortality and dementia independent of chronological age. If you would like to know more about our study, please come visit me at the poster number 15.
Good afternoon, everyone. My name is Julian Mutz. I'm a research fellow at King's College London, and I'm delighted to talk to you about my poster, which introduces a study in which I benchmarked 17 machine learning algorithms for developing aging clocks from plasma metabolite data. I used data from the UK Biobank on about 200,000 individuals and tested regularized linear and also more complex nonlinear algorithms. And I found that actually there was quite a lot of consistency across the top performing models. So in terms of predictive accuracy, it ranged from around 5 years to over 6 years. And now that's of course not what we're interested in. We're interested in how does the age delta relate to health outcomes. And I found that a cubist rule-based regression ensemble was most strongly associated with most health and aging indicators. I'd be excited to talk to you, so please come to me at poster 52. Thank you.
Thank you. Hi, I'm Ozan. I'm a PhD student with Imperial College London, and I'm interested in interrogating the environmental drivers of epigenetic aging in a UK occupation, in a midlife occupational cohort. As a starting point, I attempted to validate a range of the latest epigenetic clocks by ranking their sensitivity to smoking, as our cohort offers extensive data on smoking, blood cotinine levels, for instance, and also because smoking is one of the most notoriously known accelerators of human biological aging. So by itself acting through multiple physiological domains of aging. And so all clocks should theoretically demonstrate sensitivity to smoking, but essentially we see heterogeneity in clock performance with a consistent response from mostly the second generation clocks with current and former smokers, the relationship of which was confirmed by our analysis when I ran the clocks against cotinine. Also, smoking cessation exerts the expected reduced effects on age acceleration, as measured by the Dunedin, Pace, Grimmage, and Barnabeo clocks. Additionally, we've explored the role of diet quality and potentially modifying these associations. Poster 54 to discuss more. Thank you.
Hello, everyone. Hello, everyone. My name is Xiaojing. I'm the group leader of epigenetic from Zymo Research. Yesterday, I presented how Zymo Research latest multi-omic solution can support aging research. Today, I'm speaking on behalf of one of our substance, it's MyDNA, which offer direct-to-consumer aging test. I want to introduce our latest exploration on how biomarker of aging can benefit our pet friend dogs health. And we work with a local pet food company, which they offer a minimal processed pet food. They believe their pet food can benefit dog health, but for many years, they cannot get the age health measured quantitatively until epigenetic aging clock become available. Appreciate all the pioneers work here. And if you want to learn more how epigenetic, how you improve your dog health and get a free dog aging test, please visit my poster at 56. And I hope the organization. Thank you,
All right, last but not the least before lunch, have you ever wondered how digital devices can be used to monitor the aging process? Or maybe you want to know which digital biomarkers would actually be suitable as biomarkers of aging. To answer these questions, we've developed a structured approach for the classification, characterization, and evaluation of digital biomarkers of aging by classifying them by their physiological system, characterizing them by their capture method, and evaluating them based on criteria adapted from the biomarkers of aging framework, which includes validity, generalizability, the digital biomarkers responsiveness to interventions, its associations with clinical health outcomes, and cost considerations for usage in large-scale settings. From our systematic search, we found digital biomarkers of aging across 8 physiological systems. To learn more, please go to poster 62 on level 3. And if you want more information about me, Jessica, you can scan the QR code below. Thank you and enjoy lunch.